This is the core component Mach, and it's Holy Cross Mach 3. Question 3. Drawings show two adjacent plant cells. They look like plant cells. It's supposed to have a cell wall, large vacuole, looks like a plant cell to me. Name the parts labelled A to F, just for 2 marks. So you're going to need them all correct to get 2 marks. If you make one mistake, you're down to 1. 2 mistakes, you've blown it. Sometimes some confusion between B and A. Well, B is the cell wall and A is a thin line inside that, so that's going to be the membrane. So A is the cell membrane, B cell wall, C just stuff inside the membrane, so that's going to be cytoplasm. D and E, well, E is a vacuole, and what's the name of the membrane around the vacuole? So you could call it the vacuolar membrane, or a more technical phrase, a tonoplast. So I'm going to go for tonoplast. E, that's our vacuole, and F, the connection between that plasma desma, or you can say plasmadesmata, which is the plural of that. So plasmadesma or plasmadesmata. For 2 marks, you might have known 4 of those and slipped up slightly. Name 2 ways which materials move between cell X and Y. So materials could be anything, could be ions, water, oxygen. So for example, diffusion, why not? Diffusion of oxygen or CO2 between them. Osmosis of water. And in fact, you're looking at molar glucose solution and water potentials later on. So that should have jogged your memory for osmosis. And we've got 2 there. We have only 2 of those, but we could have the others as well. So we could have facilitated diffusion, active transport. You can start to make more suggestions, but why not just go with the main forms of transport there for 2 marks? And these cells are at incipient plasmolysis, and one chain that be visible under the microscope is placed in a 1 molar glucose solution. Right, incipient plasmolysis is this membrane here is touching the cell wall but not pushing on it, and so the cell contents, the cytoplasm, has increased in volume just enough to push the membrane, just so it's resting against the wall but not actually pushing on the wall, and so the wall is not resisting that expansion. There's no equal and opposite force coming backwards. That's incipient plasmolysis. If the cell contents, the cytoplasm, was to increase in volume, then it would start pushing on the wall, and that would be turgid. Or if the cytoplasm volume went down due to loss of water, then this membrane would come away from the wall in plasmolysis. And so we just need to work out what's going on. So we've got 0.6 molar in there, and it's saying that we get 1 molar, 1.0 molar outside. You could do this without even water, potentially just go, it's more sugary. It's more sugary outside, so water would be attracted to a more sugary environment. That's my first sort of response. And what would happen? Well, if water moves out by osmosis, then the volume of the cytoplasm would go down, and we'd start to see the cell membrane come away from the wall, the cell wall, and it would look plasmalized. So that's what would happen. So the cytoplasm shrinks. The membrane doesn't shrink. The membrane stays the same size. It's just this cytoplasm that shrinks, and the membrane will move with it. And we would see cytoplasm shrinks. You would see gaps between the cell membrane and the cell wall. So you see gaps between the cell membrane and the cell wall. So a description of plasmolysis. So it says, what would you see? What would be visible under the microscope? And now a little equation. You might be frightened by equations, but these are the sorts of equations that you've done very early on in your maths career. And then a definition. So, okay, screw the equation up, but get the definition right. And lots of different styles of this definition. This mark scheme is a really kind of expanded definition. Water potential is the kind of potential water to actually move out of a system, or water molecules to move. So that's the kind of simplest definition. So the tendency of water, I can't spell molecules to move, to leave or move out of a system. You could also talk about the free energy of water molecules. So, for example, if you've got a system with water molecules there and a similar sort of system there, if they are attached, if they are solutes dissolved in water, then there's less free energy of the actual water molecules. So the red dots have got free energy. So you could say it's a difference between the free energy of water molecules and in whatever system it is compared with pure water. But I'm going to stick with my simple definition there. Give the value of psi p in cells at incipient plasmolysis. So this is the pressure potential. So this is the force required by this wall to resist the expansion of this membrane. So if the membrane pushes on the wall pushes back. And that's just one of our basic physics, Newtonian laws. So at incipient plasmolysis, we just said it's just touching the wall but not pushing on it. So the wall doesn't need to push back, so that is 0. And here's some gaps, and we're going to have to use this equation. The first one's pretty straightforward. We've got, use that equation there, and it's all in the right order for the top one. So the water potential of the cell is the solute potential plus the pressure potential. So we're just adding these two. So you're just testing your math skills for basic equations and adding up negative numbers. So that equals, that's our equation. equals minus 1,200 plus 500. So fire that into our calculator. 1,200 minus plus our 500 is minus 700. And we don't need to put the units in here because the units are in the headings. And Q, Q, they've slightly done it so we had to rearrange the equation. So if I rearrange this equation up here, what we want as a subject to the equation is the solute potential. So we want that on its own. So to get that on its own, we need to take away the pressure potential. So this, I'll do this in just a different colour up there. So we go that, and we've got rid of the pressure potential, but we need to do that to both sides. So it's the cell minus pressure potential. So we've minused that off both sides. And so we go down here, we go that, just here's our little equation. Quite a few people just do this in their heads, but I'm not very good at maths, so I'm going to write it out. Equals the minus 300 minus minus the cell, there we are, minus 300 minus r plus 300. I think I've done that right here. So when we put that into our calculator, 300 minus r 300 minus 600. So our answer there is minus 600. There we go. So if I can do that, you can do that, because I'm a bit of a noddy at maths. And in which direction will water move between these two cells? And so we've got two numbers here for the water potential of the cells, and water will move from high water potential to low water potential. And remember your number line, 1, 2, 3, minus 1, sorry, 0 in the middle, minus 1, minus 2, minus 3, minus 4. There's our number line. And these are higher and these are lower numbers over there. So we've got water will always move from higher to lower water potential. And the highest number that we've got there is minus 300, the lowest is minus 700. So it will go from Q to P. So a little bit tricky with some of the numbers there, but there's enough definitions to keep you going there. And the last one, 50-50 guess if you can't do that. 10 marks.